All right, so this is your neuron. So this entire thing here, this is the body, the cell body of the neuron, okay, also called the soma, S-O-M-A. Soma means cell body. Now we have these protrusions that are coming off, and they're truncated. They're cut off here. We have a structure here that is coming off, another one here. This one is a little bit more noticeable up here. These are the dendrites. These are dendrites. Dendrite means branch, okay, or branches. And the dendrites function to receive information from an incoming neuron. Now in green, you'll see these other structures that are synapsing onto these dendrites. This portion, this is basically the tail end of the axon of a presynaptic neuron. The end of the axon here. So, and this is the axon terminal. So where it kind of widens, that portion of these, this is the axon terminal. Here is another axon terminal, another axon terminal, another axon terminal. Now, on occasion, you may be asked for space, okay? Usually the way that this is asked on a practical exam is they might put a sticker here or a little arrow here, and then they would have to ask you, they would have to put something like space, like what is the space? Now here you might say, well, there is no space because these are glued on, and that is true. In the body, there is a very small gap that exists between the presynaptic neuron and the postsynaptic neuron, okay? And that small little space there is called the synaptic cleft, the synaptic cleft. You might also hear that called the synaptic space, or just simply the synapse. All right. Now, the rest of this blue structure that is not one of these dendrites, this is just simply the membrane, just the membrane. Now, this yellow or beige substance here that's all within the cell body, this is the intracellular fluid. The intracellular fluid is more specifically known as the cytosol, Now, you might remember cytosol is simply the liquid inside of the cell that is inside of the cell but outside of the nucleus. All right. This is not the cytoplasm. The cytoplasm, as you might recall, includes the liquid inside of the cell as well as all of the cytoplasmic organelles. Now, this whole entire structure here is the nucleus. This whole structure here is the nucleus. Now, the nucleus here is cut open, so this kind of clear plastic structure, this is representative of the nuclear envelope, or the nuclear membrane. This blue substance that is filling this up here, this is representative of the liquid inside of the nucleus itself. The liquid inside of the nucleus itself has its own name. It's not the cytosol. Okay, remember the cytosol would be the liquid that is inside of the cell but outside of the nucleus. The liquid that is inside of the nucleus, this is the nucleoplasm. The nucleoplasm. All right. And we have two structures on the inside here that kind of look like wadded up chewing gum. One is a little bit larger than the other. Besides that, they really look identical. Okay. So the larger structure is representative of the genetic material that is inside of the nucleus. This is in its unwound form, and in this form we call the genetic material chromatin. Chromatin. Okay, so chromatin, this is the unwound genetic material that is just sitting in the nucleus. This is the DNA of the cell. We have another structure that looks just like it, but it's a little bit smaller. And this structure is the nucleolus. The nucleolus. Ribosomes are made in the nucleolus and then sent out. Right, so this is the nucleolus. So this clear coating, remember this is the nuclear membrane or the nuclear envelope. And we also have, hopefully you can, I'm going to turn a little bit just so you can see it a little bit better. There's little holes here, and now the holes in the model don't go all the way through, but you can see kind of little circles. here, little indentations. What these are the nuclear pores. The nuclear pores. So the nuclear pores are simply holes that are in the nuclear membrane or nuclear envelope that allow for things to enter and exit the nucleus. You'll notice that I have this kind of dark red structure. I've got some of it here. This is also some of the same structure, as well as here and here. This is the smooth endoplasmic reticulum. All of this is the smooth endoplasmic reticulum. Now I have a structure that looks just the same as the smooth endoplasmic reticulum. The only difference is it has these little bumps on it. So this structure here and here, this is the rough endoplasmic reticulum, the rough endoplasmic reticulum. So the rough ER appears rough due to these little bumps. These little round bumps are the ribosomes. This green structure here, so this as well as this and this is the Golgi body or the Golgi apparatus. The Golgi body or the Golgi apparatus. And you might recall that I like to call this the post office of the cell, because the Golgi body or Golgi apparatus is going to function to modify, package, and ship proteins. I have these orange structures here. So some of these orange structures are cut in half. I see one here cut in half and one here cut in half. Okay, it's just that some are cut in half and some are not. These are all mitochondria. Okay, these are the mitochondria. And the mitochondria function to make ATP for the cell. And ATP is the energy that the cell needs. The peroxisome, if you recall, is the detox center of the cell. So this is a peroxisome. This is a peroxisome. On this model, it is number 5. And then the lighter colored round cytoplasmic organelles we have here, these are the lysosomes. Lysosomes. Lys means to cut. Some, just like soma. Okay, soma means body. So lysosome, this means body that cuts. What the lysosome does is it is going to digest things. That means breaking down substances. So it acts like the stomach of the cell in that sense. And it also acts like a recycling center in some senses too, because once it does digest those non-toxic substances, then a lot of those building blocks will then be reused in the cell as building blocks for other things. So the axon is going to begin right here. Now, as this little funnel region that kind of starts the axon, okay, this portion from about here to here, this is the axon hillock. Axon hillock. Now, you might recall that the axon hillock, I have to get enough excitation, enough positive charge coming into the cell in order to depolarize this axon hillock region enough to open my voltage-gated sodium channels. And when that occurs, then we get our action potential. So the action potential is initiated here in the axon hillock. Now, once the action potential has been initiated in the axon hillock, then it's going to travel down the axon. This axon continues and it goes inside of these structures here. And I'm going to take off a little part of this here, all right, so we can see more of what's going on. All right. So this is the axon here as well. This is the axon. And then this portion has been cut away from the axon, so we can see some of these internal structures as the axon is traveling down. Now I have liquid that exists inside of the axon as well. This has a special name. We call this the axoplasm. The axoplasm. This is another mitochondria. Okay, this is also a little tiny portion of a mitochondria as well. These are very energy hungry cells. These big structures here, so these layers are the myelin sheath. Okay, so this is the myelin sheath. This is the myelin sheath. And the way that the myelin sheath is formed, it's formed in the peripheral nervous system through a particular type of cell called a Schwann cell. A Schwann cell. It's a guy's name, so it's always capitalized. It's SCHWANN. You might have thought I was just saying Schwann cell with a lisp. The myelin sheath in the peripheral nervous system is made using Schwann cells. Now, the cell body of the Schwann cell, we have a cell body of the Schwann cell that's shown right here. And so this is the cell body of the Schwann cell. The cell bodies of the Schwann cells are always going to be in this outer region Okay, so the outer region or the most superficial region of the myelin sheath. And this outer region here also has a specific special name. This is the neurolemma, So the neurolemma is number 13 here on this model. And just remember, this whole myelin sheath is made using Schwann cells. The cell body of the Schwann cell is going to be found in the neurolemma, and we have a cell body of the Schwann cell, okay? If this structure were to continue, then this would widen, this would eventually end, okay? And it would end in a structure that looks like this, all right? It would be bigger if it was to scale though, right? So, so remember, this is the axon of another neuron that's coming in, that's giving its signal to this neuron. Okay, so this would eventually synapse with another neuron, or here they're showing it synapsing with a muscle at the neuromuscular junction. And it would have structures that widen at the end, which would be the axon terminal, and then there would be a tiny gap between this neuron and either the next neuron or the muscle, and that would be the synaptic cleft again, and so on. On these models, there's a tiny space, a tiny gap between each myelin sheath. And these gaps have a name, and this is also named after a person. These are the nodes of Ranvier. So the nodes of Ranvier are simply these little gaps that we will have in between each of the myelin sheaths.